Colloquium. We're very fortunate to have Rommie Amaro come to speak to us. She's come from San Diego, California. And I'll just give you a brief introduction before we go into the talk. So Rommie has been here before. The last time you came was for, I think, BDBDB4.
Was that what it was?
Before the pandemic, we had Brownian dynamics and biological diffusion, or biological diffusion and Brownian dynamics brainstorm number 4 here. And indeed, then we had to hold it online during the pandemic, and we're holding an online version next week as well. So if you want to join us, today is the last day you can register. Okay, that's enough for the advertising. But also, of course, those of you who know Abraham know that he spent some time in Rommie's lab with an Isabel Rojas Travel Award as well a couple of years ago. So Rommie comes from Chicago and did a bachelor's degree in chemical engineering at the University of Illinois in Urbana-Champaign. And then after that, she went into industry for a couple of years and worked at Kraft Foods as an associate research engineer, apparently working on how to make Philadelphia cream cheese efficiently produced, if I understand that correctly.
How do you make it cheaper so that you can make more money for Kraft?
And then after that, she decided to do a PhD in chemistry, went back to Illinois and did the PhD with San Lucie Schulten on free energy calculations with steered molecular dynamics for an ion ammonia conduction problem in a protein. And then after that, she did a postdoc and moved to, at that point, to San Diego to do the postdoc with Andy McCammon. That's sort of where we at least share some history, because I did a postdoc with Andy McCammon, but I did it in Houston long ago, much before you. And then anyway, in 2009, she started her own research group, firstly at the University of California, Irvine, and then she moved back to San Diego in 2012 to join the Department of Chemistry and Biochemistry and as an assistant professor, and now she's moved up the ranks to be a full professor. So Rommie's work, you'll see what it's going to be about here, but she's really done a lot of methods development related to multi-scale simulations, and on the other hand, done amazing applications, in particular to look at infectious agents and infectious disease. And during the pandemic, I think her simulations of the SARS-CoV-2 virus and the virus in its environment as well were really spectacular, got a lot of press, used a lot of computing time, were really groundbreaking. I should also mention that Rommie has received many awards for her work. These include, I'm not going to, you have such a long list, I'm not going to give them all.
Sure.
But at least for the SARS-CoV-2 work, that was given, I think, 2 ACM Gordon Bell special prizes. She's an awardee of the Cohen Hansch Award in 2016 of the International QSAR Society. And most recently, 2024, she's been elected as a Biophysical Society Fellow. So without further ado, the title you can see here, Multiscale Computational Microscopy for In Situ Molecular Dynamics. We're looking forward to your talk.
Thank you. That was a terrific introduction. Right before you get started, I know that we just spent a lot of time getting the screen set up perfectly, but did you see that I was trying to click off the meeting being recorded, and then I can't find my mouse anymore. I think he's going to come down. Sorry for that. I was just trying to click that away, and then this guy came back.
Right?
But then it doesn't really let me do anything. Sorry. Sorry about that. I know. I don't want to mess it all up. I was just trying to click that away, and then I brought these guys back. Now I can't believe. Oh, I did escape, and it didn't work. Okay, thank you so much. Okay, great. Okay, thank you for that wonderful introduction. Am I screen sharing still? Actually, because I don't see the green.
Yeah, we have this.
Are you good? Okay, great. Okay, perfect. That's the Zoom. Okay, all right. Well, thank you, everyone. Thanks for that really nice introduction. It's so wonderful to be back in Heidelberg. This is such a beautiful place. I was telling Rebecca, it reminds me, I think for two reasons. It reminds me of the Midwest. It reminds me of Urbana, just sort of Germany does generally, actually. If anybody has ever visited the University of Illinois in Urbana-Champaign, even though it's in the middle of kind of nowhere in the US. It has
Flat.
it's very flat, but yes, and it's not flat here, but somehow it reminds me anyway of Heidelberg. So, okay, so I'm going to tell you today, hopefully a coherent story about how we're using simulation to do many things in biology. And I think I like to think of simulation nowadays as a new form of experiment. And I think when you're talking to wet lab chemists or biologists, this is still a little bit of a, are you sure? But I think it's okay to say that, at least that we try to, we try to hold ourselves to the same standard and actually to be equally useful, possibly, in advancing and developing models of biological systems. So, nowadays, of course, we have data coming from a tremendous number of different instruments and different sources. And one of the wonderful things about the type of models that I'm going to tell you about is that we can integrate many different diverse biological data sets taken in different labs at different resolutions, et cetera. We can integrate those data into one highly detailed three-dimensional physical model that we then make numerically computable. And so I think since this is the Institute of Theoretical, is it Studies or Sciences? Studies. I don't need to go into the details, but everything that I am talking about today primarily will be all atom molecular dynamics simulations, so once we build these sort of detailed systems, we approximate it down to the many atoms. We then define a potential function that tells us how the atoms interact. We have force fields, and then all we're doing is simply integrating Newton's equation of motion over time. So we can do that integration now, millions, billions, trillions of time steps, and achieve sampling. I mean, I think nowadays you can easily get to tens of microseconds, hundreds of microseconds, I think. And then with some strategic application of new enhanced sampling methods, we can get out to even sampling into the seconds time scale and so forth. So I think it's an increasingly powerful approach to make connections on the biological time scale, which are longer. But here we go. Okay, so we build these systems, we run the trajectories, and then we have sort of a window into the dynamics of these different systems at the all atom level. And we use that to sort of drive discovery. Can I ask one question? Can we dim a little bit the lights, or not to be fussy, but just, I don't know, can we do that or no? I can keep going. Well, oh my God, I'm making you work.
No, it's okay.
Okay, and so I like to, again, to think, well, not only simulation as a new form of experiment, but actually sort of like a computational microscope, which is not the a term that I invented. It's okay, we'll go brighter first and then we'll go softer. But one that I think is very resonant with what we're doing. That's nice.
Okay.
That's nice. That's better. You have a little more contrast. Okay, and ultimately what we want to do is really get these sort of detailed views into what I'll tell you about, into these more sort of complex, larger scale scenes, which really pushes sort of the limits of where compute is today, but I think makes some really interesting insights into these systems. Okay, and so I laugh because actually it kind of looks like a piece of liver or something, but it's actually, this is a ribosome, and we spend so much time being sort of reductionist about how we study systems, and there's a lot of reasons for that, and it's a wonderful thing that we do. In many cases, people build simulations of single proteins or protein assemblies, and then and try to understand their dynamics. And this is sort of similar to, in vitro assays that one run in dilute solutions in biochemistry labs. But ideally, what we will do is sort of actually try to understand what's happening in the in real biological context. And that's where, you know, I know a lot of folks here are interested in sort of similar, in trying to understand similar behaviors and develop methods that get us towards that ultimate goal. But that's sort of the theme of this talk today, is just trying to show you how we're approaching going from the single molecule world into these sort of more complex scenes. Okay, and so, you know, a lot of people, actually, I was just talking with, it's Fabian, right? Yeah. About why do you want to study these big systems? And it's because context matters for biology. It matters in many different ways. Even when we're looking at the structural dynamics of a single protein, context matters. I like to call this sort of in situ, of course. If you really want to bring it down to sort of a bottom line argument, it matters for drug discovery, which is, you know, a huge field where there's tons of effort and money being thrown to try at these different diseases, where of course there's tremendous public health impact. These oftentimes what we do in the world of simulation is study, for example, as I mentioned, a single protein. In this case of p53, we had initially, we and many others had done simulations of just this small little component here. This is called the DNA binding domain. And this is a nice domain because it's very tractable for experiments. If you look at the PDB, there's hundreds of different experimentally characterized high resolution structures of this small domain. We took those structures and we actually, many years ago when I was just starting my lab, you know, we solvated them and so forth, and we ran some relatively short time scale simulations. Nowadays, the kind of things you can actually get done in about less than an hour on a GPU compute. And what we found was that even sampling sort of the low-lying landscape of a single protein domain, in this case, the DNA binding domain of p53, we were able to find, we saw a pocket on the surface of the protein open up. And it's right here. And this pocket actually we showed together with many years working with many different experimental groups. We actually showed that the pocket, it exists. It's actually ligandable, so you can bind things to it. And there was a rather large effort, including a company that's now folded, to try to develop small molecule therapeutics that would target this novel site that was only seen actually in simulations. What is interesting here and relevant to this talk is that, so when we were first studying just the DNA binding domain, we discovered this cryptic druggable pocket that we saw using Markov state modeling was open about 5% of the time. So it was rather transient. In later years, we actually we shifted gears and put the DNA binding domain into its larger biological complex. This is actually part of a large transcription factor, which is p53. So this has disordered linkers and other domains, which is sort of shown here, that larger assembly, and it binds to DNA. And so when we did the simulations of this larger assembly, we actually saw a change in the conformational landscape of the DNA binding domain. And again, why we cared about that is because people have been working on p53 drugs, and they still are, in fact, for many years. But we actually saw that the pocket, which by just looking at the small system was evident about 5% of the time, it actually appeared to be more persistently open when we simulated the protein in the context of the whole, all of its partners. And that was because of specific interactions that were happening between the DNA and this protein. So we actually saw it was actually more open experimentally, which aligned with the success that we had in trying to develop molecules that would reactivate this. Okay, so it's important so the in situ complex matters for cryptic pocket discovery. And it also matters for what we call for cryptic epitopes. So you have small, again, sort of trying to get to a real sort of, bottom line. Although I will say also that sort of fundamental studies are also very important. We studied these, so you have cryptic pockets and you also have these things called cryptic epitopes, okay? And so epitopes are small segments on proteins that sort of are recognized by different immune system proteins and also for antibodies. And unfortunately, I spoke through the whole movie without actually telling you what we were seeing there. And I don't know if I can, let me see if I can go back. Okay. So we have simulated at sort of a large scale an influenza virus. And here you're looking at the atomic details simulated with all atom MD of now about 160 million atom system. And one of the things that we saw really quickly in our experiments is that, so you're looking at the virus, the red protein here is neuraminidase, the gray protein is hemagglutinin. And what we could see is actually that these cryptic epitopes that were recognized by certain immune system proteins and antibodies actually became more accessible or became accessible in these larger scale scenes. And we saw it not only for the red guy, which I'm showing you, which is neuraminidase, but also for hemagglutinin. Now, neuraminidase, what was interesting about what you just saw is that if you could see it, the head, the top part of this protein was sort of tilting. It was sort of moving around. And you could see how some are sort of flat and upright, but this one is really sort of bent. And what happens is that when this molecule bends, this antibodies actually can get access to an epitope, which is otherwise hidden in this sort of canonical structure that most people think of when they're thinking about neuraminidase, which basically has this long, it has the membrane, the viral membrane, a long stalk, and then sort of a cap on the top. And this bending actually was really important for exposing cryptic epitopes. Okay, so what I'm going to talk about in today's talk is sort of I told you a little bit about what happens when you study like a single protein and then looking at sort of a virus scale, a protein in a virus scale. And then one of the things that we've been very interested to understand, where there's enough data that we can actually build systems that are meaningful using these same methods, is trying to understand respiratory viruses and what happens to biological, just more generally, the questions that we're interested in is what happens to biological molecules when they are aerosolized. And this is a whole area of study that is in some ways is interesting because it's intersectional. You have had, traditionally, we've had aerosol chemists and sort of physical chemists working on systems, these air-water interface systems, trying to understand what sort of chemical reactions are happening at the sort of edges of these small particles. And then you have a world of biology, which is typically done, in cells and fully, condensed systems, bulk solvent and so forth. And so you have these two worlds coming together where it's difficult even sometimes to have a conversation with, across these two worlds. But in any case, what that always signals to me is that there's a lot of opportunity to learn new things there. So in any case, we're very interested in studying what happens to biological entities once they're aerosolized. And a really interesting system for this happens to be with virus-laden aerosols. And so probably most of you have heard of aerosols before. They're just small suspended particles. They can be, it's not just the stuff that comes out of like a hairspray can, which is what I guess apparently a lot of people think of when they hear the word aerosol. The aerosols can come off of crashing waves, for example, but they also are made when we speak. They're sort of emitted from us when we speak, sing, or breathe. And so some of the particles, when we're respiratory aerosols, are the ones that are emitted in this way, from our respiratory system, and they are emitted at a whole bunch of different sizes. So you can kind of get an appreciation for that here. Some of the aerosols are larger. And these actually, at some point, the terminology goes from between aerosols and droplets. So these very large particles, they actually call droplets. These are more than 100 micron in size. And so they actually have enough mass that they fall. And so this is where this whole 2 meter or 6 feet in the US, distance spacing came with COVID because they thought they initially thought that the virus, the SARS-CoV-2 virus, would basically sort of fall to the ground in the first 6 feet because they were suspended in these large droplet particles. So that's sort of what we call droplet dogma. These particles then get transferred to surfaces, and then a new word, they call that fomites. These are basically fomites are sort of contaminated surfaces with pieces of the virus that have dropped. But that's not the whole story. There's also much smaller particles called aerosols that can range anywhere. It's technically anything sort of beneath 100 micron or so, but they can actually be quite small as like 10 nanometers, all the way to hundreds of nanometers or microns. And the thing about these is they're lighter, so they float in the air. They don't sink, and they can stay in the air for hours, and then fill up a room, for example, if it's not well ventilated. Okay, so again, sort of where I was coming from with, as I mentioned, that typically aerosols have been studied by aerosol chemists who are also reductionist, like many of us, or all of us. And this is a picture drawn by the world's leading aerosol chemists in 2020 of what they thought a virus-laden aerosol might look like. And this was published in Science. This is exactly excerpted from Science. So in this image, what I think we're looking at, we definitely see the virus in there. That's the SARS-CoV-2 virus. You see it's sort of like a water droplet of some size. I'm not sure what these other dots are. I think they're ions maybe. I don't know, but that's sort of how they picture it. And this image was sort of imprinted across the globe in this Science perspective, which you can look, which has been cited, some thousands of times since then. But this is actually more like what a virus-laden aerosol looks like. It is way more crowded, way more packed with all sorts of things than that simple image that even those aerosol experts showed. And so I'm going to tell you about our efforts to construct and simulate and now analyze this large-scale system. It's a little less than half a micron in size. So we're getting all atom MD to the point where we can actually look at subcellular organelle scale type systems, and also complexity. Okay, and this system, as I was mentioning, the size that we're talking about is about 300 nanometers, so we get 350, so getting to about half a micron, and when you do that, it's equivalent to it's got about just about 1 billion atoms, a little bit more. Okay, so just looking, so what is inside? So how do we know what's in that system? Actually, there's a whole, in aerosol science, there's, a whole group of folks who are working on aerosolization processes and sort of to understand what gets transferred from the bulk fluid into the air. And I have to say the mechanics of that process and what gets transferred is also fascinating. So in our lungs, inside of our lungs, so this is down here sort of would be the surface of our lungs. Our lung is an air-water interface, of course. There's lung surfactant in there. There's all these surfactants. There's these things called mucins, which are amazing molecules. I'll tell you, well, actually, yeah, I'll talk a little bit about that later. There's all sorts of bits in there. There's also lipids and ions. And what happens is, you have all of these biological things in the lungs. And then when you exhale, you form these film bridges. So as the spaces sort of fill up with air, you get these air pockets, and then you also get these bridges, and then they burst, and then you end up with these aerosolized, these particles. They're many different sizes, and then sometimes these particles actually, because if you have virus in your fluid, it will actually, that virus will get transferred along with things like mucins, proteins, and other lipids to create these sort of more complex mixtures than what they showed in that original picture. Okay, so these aerosols are produced by a process called film rupture. And what we're interested in, just scientifically, is really, I think it's fascinating what happens to biological things in the air. when I first started working on this 5 or 6 years ago, or in 2018, I guess that's even more than 5 years ago now, my gosh. When I talked to my biochemistry colleagues, they were basically like, Rommie, why are you even studying this? It's going to get in the air. It's going to get destroyed. Like, there's nothing interesting there. I'm not going to do, because I was trying to convince them to run some experiments on these enzymes to find out if enzymes were also actually active in the air, which in principle should be measurable. Anyway, they kind of poo-pooed me, but which is fine and understandable. But there's all sorts of interesting questions here, because yes, some of, when biological molecules and entities are aerosolized, absolutely, the air environment is very destructive. Okay, we know already from, if you're a structural biologist and you're putting something into like a Titan, one of those wonderful Titan microscopes, the air-water interface kills your molecular structure. But somehow we also know that for sure, for SARS-CoV-2 and other viruses like measles, they are airborne, and that means that they are staying viable somehow when they're aerosolized, and to different degrees. But like, we really don't understand much about like, why is that the case for some viruses and not others? And it's not only the air-water interface, but there's all sorts of really cool chemical reactions happening in the air. If you're indoors or outdoors, it's different, because you can have reactions with sunlight, and you can also have reactions at the actual interface of the aerosol with different atmospheric gases and stuff. So in principle, the atmosphere is very destructive. But somehow these viruses actually can't, some, not all of them, but some and enough, actually can stay aerosolized and then be breathed in and actually infect someone. So we're interested in many things on the basic science side, trying to figure out, to use these simulations to understand what are some things happening there. Okay, and so how do we go about doing that? So, okay, you have this complex mixture of a bunch of stuff. Like, where do we start? Well, we are still reductionist, and our approach is, once we know the like molecular ingredient list from, for example, mass spec of these aerosol particles, then we basically start at the top, and we have a list, and we try to make sure that every ingredient in the list is going to be able to be simulated, basically, right? So first, of course, because we're interested in virus-laden aerosols, we started with the virus. And to even start for simulating a virus, the main thing that we needed to start with was the part that actually infects humans, which is the spike protein, which is on the outside of the virus. So we start with the spike protein. And so this is the workflow. I'm not going to walk through it in detail, but this is just to give you a sense for, if I can say, sort of how hard we work towards making sure that these are high quality models that are aligned with experiment. So every part of these bigger scales, larger scale systems, ideally, every part of that system will be a high quality model that can stand up to experiments. Because in the end, at least for now, we still do measure ourselves with the benchmark of experiment, or what new hypothesis have we made, that helps us understand a system, usually through experiment. Okay, so this is, so this means that we start with some usually for these systems, this is a fusion glycoprotein, the spike protein on the outside of the surface. We start with some amount of structure is usually resolved by single particle cryo-EM, that's sort of in the top middle there. And then we have to fill in all the missing things, right? And so as amazing as structural biology techniques are, even with these fantastic microscopes, and there's science paper after science paper after science paper with new really amazingly resolution structures, they still can't see highly dynamic parts, right? So they can't see, if usually they can average to a high resolution, the ectodomain, which is shown there in the middle, but the stalk, which you can see on the right, there's all these parts that we have to build through different types of modeling procedures. It could be homology modeling. It could be something like alpha-3 now, AlphaFold 3, et cetera. Once we build the structure, another key part that's missing structurally are the glycans, these end-linked glycans. And so for the glycans, which are sort of these brightly colored little bits, they look like ornaments coming off of a Christmas tree. We know from mass spectrometry, they can get the composition of each of these glycans. And so then we can build those, we can actually reconstruct these glycans. We can also take care of the membrane parts and build in post-translational modifications. These are palmitoylated cysteines. And here's our membrane with some different sort of molecular features to it. Okay, so we get all of this. And then we're able to, is this going to wiggle? Oh, no, this is a picture. I wasn't sure. Okay. So then we're able to actually get a high quality model of the protein. We published this one of the SARS-CoV-2 spike in 2020, and I think it's been pretty useful. So as I mentioned, each molecule in the scene will hopefully be a useful model that's experimentally validated. And I'm just going to take a short segue here to say what we found for example, and I won't do this for every protein that we have in the respiratory aerosol, but this one, I'll just say what one of the things simulation showed us was this glycan shield. So on the left is basically essentially what the structural biologists see. They see the protein. And on the right, these blue sort of fuzz that you see there, those are the end-linked glycans. And you can sort of, that's a composite image of their dynamics over 1 microsecond. So you basically see how these glycans sweep out in effective space around the molecule that basically hides the molecule from the human immune system. En route to this, we also discovered there were 2 glycans, which you can see here in sort of van der Waals representation or space filling. There were 2 glycans that basically position themselves to help keep the spike in an infectious conformation, which is when the RBD, which is when it sort of has this exposed receptor binding domain. And this was also, so this was nice because it not only sort of advanced our understanding of the spike protein, which, you know, in 2020 people were really interested to do, but also because it discovered a whole new role of glycans in biology, which is that, I mean, most people were thinking about glycans being just shielding of the immune proteins, but actually they were shown here also through experiment to actually play a role in the infection mechanism itself. And that's now been shown also for other viruses. So that was kind of fun. Okay. So we have all these different proteins. And again, I don't want to go through all of them, but that was one example. The next thing that we have to do, and actually one of the hardest, is anything that has to do with a membrane. Membranes are like wonderful. They're wonderful. And they're also terrible. And but of course, they're really critical to understanding biology. And I think still one of the biggest hurdles into simulating at like cell scale with any appreciable detail. So we and many others have worked on different methods to construct membrane models at scale. One of the ones that we still use, which was developed many years ago now by Jacob Durant, who's a professor at the University of Pittsburgh, is something called lipid wrapper. And basically what it allows us to do is we have these, you can equilibrate sort of a membrane patch of any composition that you want. And then if you any surface that you have, which could be just drawn or could be taken from cryo-ET, you can actually just tessellate that surface and then insert these patches into that surface and build these different, in this case, most of these things are circular because it's like the viruses might not be exactly spherical, but they are quite spherical. So we start with that. Anyway, so you can build those. And this is one of the issues. And this is Klaus Schulten, who is just wonderful, who was a fantastic mentor to me. And he told me one time, which is true, and us working with others, is that, you know, if you have a small system, a small protein, you sort of have small problems. If you have a big system, you end up with big problems. And you see this with membranes and big systems, is that you can have a really nice looking model, but if there's a little bit of an inaccuracy in packing, for example, in a small system, okay, it's like one small problem. But when that small problem in a big system is replicated 1,000,000 times, you now have a nightmare on your hands. And this is exactly what happens with membrane building, that you have, it's just very hard due to the curvature of the inner and outer leaflets to get a numerically stable membrane. One of the things that you could actually see in this slide was Oh, well, there were like holes forming. So we build these membranes and then you'll have holes basically forming and you have to fill the holes and patch them. And it's really a process. So we've been working on how to, I think part of my, the D is off. Yeah, it's fine. It's covered. So we actually now have worked on a tool. This isn't published yet. It's just in preparation. We've been working on this workflow, which we call the Dyson method for, I won't go into details because it's too detailed. But basically, we've been working out now, we have worked out a way to much more rapidly build these large scale membrane models that are numerically stable at scale. And it just basically involves a bunch of like small technical details, but we've now made it into a workflow that sort of it processes a lot faster. And so we've done 6 different systems with 20 different lipids, and even now including glycolipids, which are cool. So we have, and again, this is stuff, this should be submitted sometime this next year. But we were looking at HIV membrane, plasma membrane, endosomes, exosomes, synaptic vesicles, and we're able to create these large scale models much more rapidly. And I'm really excited too about these glycolipids because you can see it'll be interesting to now have those for relevant membranes, to have those also in there to understand things like diffusion and interactions with the different molecular components. Okay, so that's the membrane. Then how do we build the virus? Well, we actually turn to more experiment here, in this case of SARS-CoV-2, but in many of the most of the structural biology, we're using then cryo-electron tomography, which gives us lower resolution, basically molecular maps that show us the pattern and density of where we want to build in these high resolution pieces of these spike proteins. And we can also use the measurement of the virus here, of its membrane, to build the correct viral membrane. Right, so the important takeaways from these types of experiments is they give us the virion diameter, and they tell us, in this case of SARS, actually, the spike protein density is pretty low compared to things like influenza, and also that they're sort of randomly distributed. They're not like uniformly distributed on the surface. Okay, and so then we build this. This is what the sort of fully constructed system looks like. You have the membrane, which is shown in purple, and then we have inserted, and I don't go into details, we have the E protein and the M protein. These are just structural proteins that sort of sit inside the viral membrane. And then you also have that model I told you about of the actual spike protein with all of its fun things. Okay, so we did the virus. Then how do we pack the respiratory components? Well, we know, again, from mass spec, they sort of have a pretty good idea of what's in respiratory fluid. It does actually change a lot depending on who is breathing, which is interesting. The other thing that's interesting to me is that we're all, like, I'm up here talking and y'all are breathing, and with the exception of the very smart person there who's wearing a mask, very nice, you're all breathing in the stuff that I'm putting out, which is kind of interesting. We share the air, including all these little molecular bits, which is a reason why it's could be very smart sometimes to wear masks. But in any case, so inside our respiratory fluid, there's lots of different ions. There's lipids free floating in there also, including DPPC, cholesterol. There are albumin proteins. You can see some of them there. And then there's these other amazing molecules called mucins, which is a whole, we had to basically, to get to this level, we had a lot of publications on the virus, but we also have publications on mucins because that was sort of a new thing that we had to develop. Okay, and then we put it all together. How do we put it all together? There's new methods now actually coming from the group of Mary. He has a really nice new packing thing called Bentropy, I think. Is it Bentopy or Tropy?
Bento pie, like a bento box.
Oh, like a bento box. Ah, okay, I like that. Okay. And now I know how to say it. It's bento pie. Anyway, initially we just used Pacmal. So basically Pacmal just lets you like randomly pack a bunch of ingredients into like a cube. So we did that, and then we did it for a smaller box that we then could like actually equilibrate and sort of like get to an okay positioning, because otherwise it's just literally randomly packing these ingredients. And then we replicate that. This sort of looks like a Rubik's Cube, but we just replicate those different boxes together. We stitch them together. And then we have to actually cut out essentially sort of a sphere of that and insert into it the virus, which you can see there. Okay, so this is just like big system fun for those of you. I think I'll just say again, another sidebar. I do not, I want to say as much, I obviously I love doing MD of big crazy systems, but you don't necessarily want to do this for every system, right? I mean, it's very expensive, and probably we definitely need to be more targeted, and multi-scale is the way to go for sure, including Brownian dynamics and so forth. But the story I'm telling you today is all about all atom MD. Okay, that was my sidebar to you all here, because I know there's a lot of folks working on multi-scale, which is really amazing. That's where we have to go. But in any case, you can sort of see the scaling. So when this system is huge, right? 1 billion atoms with all atom MD is like, where are you going to run that? So we actually were able to run on the biggest machines that we have access to pretty much in the world. There might be bigger ones, but the ones in the US are in the government machines, like the Department of Defense and the Department of Energy. And so we were able to actually take over half the machine and then the whole machine for small bits of time. And so we could actually get scaling. So this is a 1 billion atom system on 256 nodes on Frontier, I believe this is. Nope, this is Oak Ridge, which is the machine used to be called Summit. It's been decommissioned now. On 256 nodes, we got about 4 nanoseconds a day. And if we scale to the whole machine, we could get 34 nanoseconds a day, which is slow. But it at least gave us, we were able to do it. And the other thing I'll say too, I'll just sort of preempt the question that might come later. You're like, what are you going to learn in a couple hundred nanoseconds or like a microsecond? Like literally, what are you going to learn? And I will tell you that these questions in aerosol chemistry are they are aligned with that time scale, because the mixing of components in the air in these small particles, actually, you can see that you reach an equilibrium after like a few hundred nanoseconds. That's probably not the case of necessarily like cell scale scenes. But here are questions, I think we're relatively sort of well aligned with it. Okay, so this is what the system looks like. You can kind of see here, if we could dim a little more, you would see, but these guys are wiggling and jiggling. The atoms here in green, I think they must be ions. We're not showing any water molecules, and you can see the virus and the spike proteins. And here's a zoom in of a spike protein in light blue. These are the glycans in dark blue. The green guy up there who's interacting, that's an albumin. You can see a free-floating lipid there. The lipids also do interesting things. They just kind of like, some of them glom together, some of them actually just go to the surface of the protein. And then these red guys here, sorry, it went away, but the red guys, those are mucins, which are these polymer-like molecules. They're O-glycosylated protein backbones that sort of like have a bottle brush sort of shape and dynamics that are very important for modulating sort of the physiochemical properties of air-water interfaces in our body in different mucosal membranes. Anyway, okay. Again, so why do we care? So similar to what we found when we looked at the protein dynamics of the glycoproteins in influenza, we actually see that, so this is now looking at okay, let me also just pause here. I think it's okay to pause. This movie's just gonna go on and on. There's so much to analyze, right? Think about if you just have like one molecule and there's a lot to analyze, a molecule with a membrane, there's a lot to analyze. Now you got a lot of molecules. Like it's, I think one of the interesting things about this, it is a place where sometimes also just like your human intuition breaks down, even sort of thinking about how to do this. But here so we've tried to do our best to analyze what we think are some of the more interesting features, but it's just scratching the surface. So the spike protein, as I told you about, a lot of work has been done to understand the dynamics of the ectodomain, which is that big top blob with the receptor binding domain being up or down and various mutants, et cetera. But it also has these other sort of dynamic hinges connecting the stalk to the membrane. And so what I'm showing here is the distribution of different tilt angles taken over the course of so many microseconds of data for the single proteins. So if because as I mentioned, we had a lot of microseconds of dynamics of just a single protein, and we get a distribution of angles for the hip and the knee, which is kind of in the middle, and the ankle, which is close to the membrane. And then when we actually, we can look at the distribution of angles sampled in our complex scene, and we see we have a right shift for the hip, for this, you can sort of see a slightly different distribution of states for the hip angle. And same with sort of the knee angle, which is in the middle. And then the ankle actually goes the other direction. But so here we're not looking at cryptic epitopes, although we could. Actually, I probably should. But even preliminarily, we can see these differences in the conformational landscapes actually sampled of the molecules compared to just the dilute solution sort of in vitro MD simulations. And then the other thing that, I mean, one way we're thinking about doing the analysis is that to create sort of an interactome of different protein, of different things, of different molecules for each component in the system. So we can look at the spike interactome. So this is basically telling us what is the spike interacting with over the course of this simulation. It's interacting with just free DPPC molecules, a lot with mucins, and the sort of the size of the bar is telling you the propensity, the sort of relative propensity of interactions. We have a lot of interactions with the M protein, which I didn't tell you about, but that's basically, it's basically associated with the spike tail on the inside of the cytoplasmic tail and the inside of the membrane. It's associated there. And then calcium, albumin, et cetera. And we can do it not only for the spike, but for the albumin and other components of the system. So we can already begin to see like, well, what is it interacting with? The other thing that we're interested to do is look at like diffusion inside of this guy. And so here, sort of what I'm trying to illustrate is we have our aerosol, and he took Nick Lauer, who's this terrific grad student who's going to defend when I get back. Actually, the day I get back, he's going to defend. We took this slice out of it and then laid that slice like a slab. And you can see that the diffusion constant, even of water, is like heterogeneous in this system, right? And so in the middle there, the diffusion constant is high, that's of water. And so it is, there's nothing inside of the virus, I should tell you. The virus does not have, it should be packed with RNA, but we did not even pack it with RNA. That's like a whole nother set of talks, how to pack that thing. We're mostly interested with questions about what happens to the molecules on the outside of the virus. So we allow ourselves to make that approximation. But then you can also see how the diffusion of water changes depending on what it's near to, like mucin, spike, and albumin. So you can kind of make that mapping between the diffusion and the actual components in the system. And so one of the things we saw was that mucins and the spike protein and albumin, they actually slow diffusion of water because they're beginning to interact. So that makes sense. And then we can just look at diffusion of the actual, not just water, but the actual molecules. And one of the things we see, so this is the virus in the middle, and you can kind of see it here. The contrast is kind of okay. Fast diffusing molecules are shown in yellow, and you can kind of see them. I think those are mostly albumins that I'm displaying. And then slow diffusion are darker colors like purple. You do have, in general, higher diffusion of molecules closer to the surface. You have slower diffusion when it gets close to the core. And we also see the influence of aggregation. So the other thing that we're interested in looking at is like, what species are there aggregates? What species are aggregating? And then like what kind of properties change? And we can see that in general, proteins that do form aggregates, like the mucins will clump up and albumins can clump up together. Clump up, that's very, that's my jet-lagged scientific lingo. When they form aggregates, they also tend to exhibit lower self-diffusion. And then, right, so we can look at diffusion at different timescales. Like I showed you before we were looking at diffusion in the aerosol of albumins and mucins. We can, of course, also look at how the spike protein even diffuses inside just of the viral membrane. And again, this is colored by displacement. And then even further, we can look at, okay, what's happening to like the lipids in the membrane. And you can see here, looking at the displacement of these different lipids, I forget what they call this technique, but it's basically drawing sort of lines, sort of showing the directionality of the lipids. Anyway, okay, so we looked at all of that. And then proteome aggregation dynamics. So the proteome of the aerosol, we do need to look for aggregation, and we can actually see these different aggregates and what other components are forming these aggregates and at what propensity. So we find albumin mucin aggregates at some propensity, albumin aggregates, straight up mucin aggregates, and then different aggregates with the spike. Okay, so you can sort of, again, we have to sort of bring these tools together to sort of understand what's happening. And then once we understand the aggregates, we actually use a graph theory analysis and the diffusion to show how these different sort of clusters of proteins and their movements are sort of correlated or connected. Let me see, what's the most important thing? Because there's a lot of words here. I already said most of this. In essence, because I know at this point it's late in the talk, but I'll just say that understanding the relationship between aggregation of molecules and their diffusion has implications for understanding how these droplets and aerosols, how their molecules sort of evolve over time, and which, as I will get to, is also important for us understanding of what happens to the virus in these complex environments. Okay, and so then taking it back to like, what are we learning about the biology here? But before I say that, I want to say from the aerosol chemistry side, one of the things that's interesting about these studies is that there is no experimental technique that can inform on the atomic details of an aerosol or droplet structure. It doesn't exist. So aerosol chemists were excited to actually see like, well, what could possibly be happening? Which molecules are going to the surface to participate, for example, in chemical reactions with these atmospheric gases? We are interested more in general, I'm more interested from the biology side. So what's going on with the virus? Okay, this is a picture, another rendering of that same aerosol I've been showing you. This is actually packed with the new program, Bentopi. Is that how you say it? Okay. And you can see, so the red molecules, those are the mucins. I mean, it's insanely packed with mucins. All right, and it's like bumping right up against the structure. So what we think is that the virus is obviously very well packed in these respiratory aerosols with mucins, and the variants of SARS-CoV-2. Remember, we had the first wave, which infected everybody, and then there was like the Delta wave, and then there was Omicron, and it keeps mutating. One of the things that we've learned is that the biophysical properties of the spike proteins is evolving with these variants. And so if you look at the wild type protein, I call this wild type. This was the first strain that folks were infected with in like 2020. If you looked at the formal charge at pH 7, it was -11. So here I'm showing, negative is red and electrostatically positive is blue, neutral being white. It was -11 in the first wave. It was +4 in Delta. It's +10 in the first Omicron. So it's getting considerably electrostatically positive. And what we see is that electrostatic positivity is sort of remapping into concentrated areas. And the positive charge on the spike protein is very complementary to the negative charge of those mucins. So the mucins are negatively charged. And we can, I don't even put the paper here, but we've showed that things that are molecules that are negative, actually they associate faster to these variants. And in the case of the aerosol, we think that these variants are basically evolving to interact better with mucins in the aerosol, which is why actually, if you look at, they've looked at infectivity experimentally, they've looked at the infectivity of the different strains in aerosols. This is work done at a wonderful aerosol center in Bristol, in England. They actually see that the aerosols are in fact more infectious over time. And we think the virus in aerosols, as they've gone through the variants, is more infectious over time. And I think it's because it's basically they're getting more protected by mucins. Because as the mucins come and interact with the spike protein, they form like a protective kind of gel, if you will, around the virus. And then that virus won't get exposed directly to the air interface, which otherwise would completely destroy its structure. Okay, that's hypothesis one. And then the second thing I'll say, and then I'm definitely done, is that that same aerosol center under the leadership of Jonathan Reed at Bristol sought to understand how ambient carbon dioxide affects aerosol infectivity. And so you have to put your PChem hats on, which I think is always fun, unless maybe you don't take it off, I don't know, or maybe you don't have a PChem hat, but here, we'll put it on. So, in the lungs, you have high amounts of carbon dioxide. And so when the viruses when these aerosol particles are made, they have a lot of CO2 gases dissolved in them. And then when we emit them into ambient air, it off-gasses CO2. When off-gassing happens, the pH, because of bicarbonate, the pH basically, the pH undergoes a rapid increase. It becomes much more alkaline. So pH can be in these aerosols as high as like 12 or 13, which you're like, to me, I hear that and I'm like, what's happening biologically at that pH? But basically, when that happens, if you can imagine, if you're a biological structure and you are all of a sudden at 12 pH, you're very highly negatively charged. And so all the points that I show you there that are gone from like basically purple or blue to red, those are the sort of negative charges that are introduced at pH 12. And immediately, so we've simulated under these high pH conditions, and immediately what you see start happening there, this is just a patch of the membrane. You can see that hole immediately basically start to open up. So we think actually one of the major deactivation routes of the virus at high pHs is that one of the first things that happens that kills it is the destruction of the membrane. But, okay, hang on. So if you have a poorly ventilated space where a lot of people are breathing, this is, by the way, wonderfully ventilated. I remember when I came here before and measured it, I didn't bring my CO2 detector, but it's probably nearly outside carbon dioxide levels. But in some rooms you're in, if you have one of those monitors, you'll see, and in planes, for example, it goes up to like 3,000, 4,000, where ideal outdoor conditions would be carbon dioxide at like 400, maybe 450. And so that's a signal that you have a lot of air that's being rebreathed by you and others, for example, in planes. And then if you have high carbon dioxide in the air, that off-gassing doesn't happen. And then it basically means that high CO2 environments, like in these poorly ventilated spaces, actually create more cell-like conditions because it keeps the pH low, and that keeps the virus sort of viable in the air, which is why you should wear a mask, which is really smart, and open the windows if you notice that it's getting stuffy in there. Okay. All right. With that, I'm going to stop and I'm going to put you out of your misery. Sorry, that was really long. I will say another, as you can kind of tell, big systems involve a lot of people. In my group, when we're building these, it's not uncommon to have different people sort of own a different part of the system. And each, for example, grad student who's studying protein X, protein Y, protein Z, or postdoc, they get their own set of publications just on that single system. And then we bring it together into the whole. So, and it takes a lot of folks. So this is just a thank you slide to everybody. And then I'll also say thank you to you for your attention. And I'm happy to discuss or take questions if we have a couple more minutes.
Okay, thank you very much. Really fantastic talk. I mean, one just has to look at these and it's never boring, right? Look at all of this. So there's just so much to learn. And it really shows what you can also get out of simulation. So we do have time for some questions. If you want to ask a question, you should come here.
Oh, I guess that's for Zoom.
Yes, that's for the
there you go.
People far away. So Fabian.
Yeah, so thank you very much for the wonderful talk. I have two questions, but let's start with one on the aerosols. So one thing you said is that, you can simulate them for hundreds of nanoseconds, but then equilibration also happens at that timescale of processes. And could you elaborate a bit more on what the evidence is and how you maybe measure equilibration in those systems?
Yeah, so, well, for us, we measure equilibration, and that's a good question. It's sort of like when the dynamics of the different components of the aerosols, at some point they basically, you randomly pack them and they move around a lot, and then they find their aggregation partners, and then they also, some things will move to the surface and some things will sort of stay in the bulk. And that process all those processes kind of tail at some point. And I'll say, and I didn't show this here, but in earlier work, or other work, we also studied a different type of aerosol called sea spray aerosol, which is just coming off of crashing waves, where it has also biological components. It doesn't, in this case, it didn't have the virus, but it had a lot of the other things, and fatty acids and all this. And we measured sort of how the morphology is changing over time, and also the dynamics of the different components in the system. And it tails off. So that's how we sort of measure it from a simulation standpoint. And then in terms of what are we seeing? Is what we're seeing actually, is it just like an artifact of simulation, or is there some kind of truth there? Those questions, we typically we try to make a hypothesis around what we're seeing and then test that experimentally. So that could be reactions that are happening. So some molecules will have a higher propensity to be more surface active, going to the surface. And then they could react with atmospheric gases more, or you can measure using different experimental physical chemistry techniques, like what's on the surface and so forth. And then for the virus, we can look at things like infectivity and so on. Sorry, that was long-winded.
No, that's fine. Yeah, very interesting. And then if I may, I continue with a question for the SARS-CoV-2 nerds.
Yeah.
So we have studied, a while back also the viral envelope of the SARS-CoV-2 virus, and one sort of experimental detail that always struck us was the concentration, or the supposed concentration, of the M protein in the surface of the SARS-CoV virus, which is really, really high, even compared to biological membranes.
Yeah.
So I was wondering if the simulations that you run, are they actually at this high M dimer concentration? And do you see this lattice formation that people are now reporting experimentally as well?
Yeah. That's a really good question. So Fabian's question is about the M protein, which I showed you super, super briefly. The M protein in all the viruses that I showed, I had the membrane in purple, and then you might have seen a lot of little gray bits. That's the M protein. There's like 1,000 copies of the M protein. It's called the matrix protein. And so you can kind of see little bits of the gray. That's the M protein. They can't structurally resolve that. John Briggs is working super hard on trying to resolve the matrix protein. It just is really hard because it's inside of the tomography resolution it just gets too fuzzy. So It does. So yes, so we actually worked very hard on trying to come with models of the different arrangements of the M protein dimer. And yes, we chose one of those to model into this system. So we have that matrix. So the matrix protein is, they call it a matrix because it does form like a lattice inside the viral envelope, and it does give the viral envelope like some stability. We do have that. But it's just, I think still even to this day, although I should probably check, John is an amazing experimental structural biologist, probably he'll figure it out soon. But we don't have any direct way to compare if, how we've modeled it yet is the right, we don't really know exactly which form it should take necessarily, but yes, we have it and we chose one, which we hope is right, but it may not be, but we don't know yet.
Okay, then thank you very much for the answer.
Thanks.
We'll talk more later.
Okay.
More questions, Ricardo?
Yeah, you can come on. Don't be shy.
Yeah, thank you very much for the talk. It was a very nice journey. So my first question is, how do you model the outside of the aerosol? So do you have just a continuum dielectric? Do you have like any other energy terms? Or
well, for
it's just vacuum?
It's a vacuum. And so thank you for asking that because I didn't say it, but because we were using NAMD, by the way, to do the big simulations. We still find NAMD to be the best code. So basically you have your sphere that has, normal bulk, and then you define your periodic box like outside of that. And actually I should say, but I didn't, although we talk about it a little bit, I think in the Gordon Bell paper, because of the way NAMD is parallelized, it actually, when you have a box that doesn't have a lot of stuff in it, the communication between the boxes that have stuff and not have stuff in them actually is one of the things that kind of slows it down. So they had to do some special tweaking to try to optimize that. But in this case, it's a vacuum. And you will see, by the way, maybe this is going to be your next question. In all of these aerosols, you do see water evaporation, which is like the water molecules actually like leaving the thing, the aerosols. And this can, in some instances, be compared to experiment. But did you have another question?
Yeah, it was unrelated. But the second question was on the way that you define the interactome for the molecules inside the simulation. So I was just curious about how
do we decide?
Yeah, it's just like pairwise. You have just some pairwise interaction and you define like an interaction. And like related question is, do you have somehow an experimental validation or comparison for those interactome, or it's impossible? Okay.
Yeah, so yes, we do like a distance calculation over like all the molecules in the thing. And I forget even what we have as the cutoff for interacting, but I can look it up. Yeah, so that's one part of it. And then once we make the interactome, at this point, until they get structural data, experimental structural data, like of aerosols, I'm not sure how you would actually experimentally say, oh, this is the interactome or not. So, yeah, I mean, definitely we're still in this position. It's kind of a weird position which is fun, but also sometimes difficult because there's places when you have, there's like projects where everything that you're studying pretty much can be directly compared to an experimental observable. And then at some point though, you get into a realm where we don't have the experimental ability yet. But what I'm hoping is that because there's so many brilliant experimentalists all over the place, that they eventually develop the technique, and then we can come back and say, did we get it right or not? Or what could we change in the model, to make it more informative? Yep. So it's an interesting space. Okay.
Thank you for the intriguing talk. It got me thinking for the first time about the mucins and how they interact, which I thought was very, very interesting. especially if you compare it to the process of like lyophilization. And that's also what you started mentioning is the water evaporation in the vacuum simulation. And my next question is like, if I mentally model a virus like droplet going out from the lung, then after a few seconds, it should start really drying up.
Yes,
like,
it
the
also
vapor
depending
pressure
on the
should be
humidity.
extremely high because of the small curvature of the
Yeah.
droplet. So is this something you're going to look into? Is this like do you think that you can simulate this like maybe instability or like protein
The dehydration process?
Is
Yeah, that's a good question. In fact, so in principle, yes, but it probably would just take a lot of time, and you have to also, so the other thing that I didn't tell you is that we set a relative humidity, like the water concentration that we have is determined at a certain relative humidity, like of the environment. And so if you have something very dry, it evaporates really fast. If you have more, in principle, that can be, we don't do it. I don't think it's something that we're actively doing, but there have been a couple groups who've done that, and they do it, gosh, they have some pretty ingenious ways to try to do that at the atomic resolution by sort of manipulating the system setup over different conditions also. I can try to send that to you if you're interested. Yeah.
Because it also interacts with the point you were making about the pH.
Yeah.
Because as you evaporate out, you increase the pH even more.
Yeah.
Like
exactly.
As you exponentially as you reduce the radius. So can you snapshot this? Do you think is this like a?
Oh yeah, you can definitely do things like snapshotting. Yeah. We have not done that. Yeah, but it's probably that would be one way to do it.
Oh, thanks.
Yeah, you're welcome. Oh.
Okay, there's one online question. I don't know if you can read it on your screen, otherwise I'll read it out for you. So it's from Pachwal. So glycans are helping to keep spike head in active pose upright. Are there other components present in the simulation system that have the same effect?
Good question.
Any observation during the MD? Is there experimental validation for the same?
That's a great question. And you know what? I actually am not. So when we did, that's a really good question. And it makes me think maybe that's something we haven't analyzed that we need to stick into the paper before we get this out. Or did we analyze it? So they're asking if the sort of structural dynamics of the RBD in terms of like open down, if it changes. And I also want to take this as an opportunity to say that when we first construct the virus, we don't insert the exact same spike in all the places where it is. So maybe let's say there's 30 spike proteins on a virus. We run the dynamics of a single spike protein, and then we cluster the snapshots, and then we insert slightly, we insert, they're sort of similar structures, but they're not identical. And we also know from the tomography experiments, for example, of Beck and Briggs and others, that they could see already in tomography that there was some number of spike proteins of the 30 or 40 spike proteins in the virus. Some were open and some were closed. So in our initial virus simulations, we actually had, I don't know the numbers, maybe we had 8 closed and 24 open or something like that. So in principle, we could look at this also to see if there's variation, but to be honest, I'm not sure. I don't recall if we've done that, but that's an interesting question. And one would think that also just sort of following that further, when you have the mucins packed onto the virus, if you have a mucin laying on top, does that help to keep it perhaps closed?
That was what I was wondering, really, because if you're saying you've got all these mucin on the surface, it's okay when it's sort of floating around as aerosol, it's fine. But at some point, it's going to have to infect the next
it's going to get
host,
rehydrated.
and it's going to compete with all that negative stuff on the proteoglycans and so on.
Yeah.
And so the mucin
And they
has
do
to go
bind.
off. Do you look at that?
We have not gotten to that, but It is sort of further down in our thought experiments, I'll say that, yeah. And we have also done a lot with the host cell proteoglycans and trying to build molecular models. And we know from work we've done that, oh yeah, and you also, looking at heparin sulfate and things, how it binds and actually could help encourage it to open or keep it stable. That's maybe too much for my jet lag mind to continue to elaborate on, but yes, we should do that.
I understand. Okay, I think we have one last question, Fabio, right?
Oh, yeah. Are you coming in with a lipids question? Oh, yes. This is also yeah. Oh my gosh. Let's go for it.
I think it's very fascinating, the level of detail that you apply for the, to the protein. My question is that, well, on the SARS-CoV, did you also analyze exactly lipid behavior and maybe close to the spike proteins or far away, and if you see any difference? That's one. And the second one, I mean, it's somehow correlated, but how do you choose the composition of the membrane?
Okay, cool. Those are two great questions. I'm going to answer your second one first. So for the composition, the way that we approach this for all of these types of systems is we just go back to the biology of where the virus is made. Some viruses like HIV come off of the plasma membrane. And so then we try to do a best first order approximation of the plasma membrane. In the case of SARS-2, it buds off the Golgi intermediate compartment. So it's a little bit different. So depending on where the viruses bud, they have slightly different, and that's what we try to match. So here we tried to match again to a first order approximation, which is like maybe we have, for us, first order is like anything where it starts to get really small, but typically it has maybe like 8 to 10 lipids, different phospholipid types. And then we've not really done an analysis of particular enrichment of lipids. And that's just because there's just so much to look at. It's one of those questions where I say, yes, we should do that. Or what I'm more excited about is it's just like, we definitely can't analyze all the data in this system. And there's questions that people are going to be interested in. We're like, I have no expertise. And so one of the things we're looking forward to is sharing the data. And so then people can go in and just see what happens to the lipids. Are there enrichment of particular ones near the tails, or what happens to the different post-translational modifications? I mean, there's so many different questions one can ask. Yeah.
Thanks.
Okay, well, I think we have to finish at this point. So thank you very much. Really fascinating talk.